this presentation will be coming in two parts. The first part will be with me and the second with Adam Clancy. So in my part, I'm going to be giving what I would think is a flavour for what organic chemistry is and why it's important, and also a flavour of the sort of ideas and concepts you'll be exposed to on an undergraduate organic chemistry course. And in particular, I'm going to be focusing on two short stories on how our understanding and application of organic chemistry is of critical importance in the drug and pharmaceutical industry. And then in the second part with my colleague Adam Clancy, he'll be telling you about his enthusiasm for nanomaterials, and in particular 1- and 2-dimensional nanomaterials and some applications of carbon nanotubes. So very briefly, to introduce myself, my name is Alex Fawcett, and I am a researcher in the Department of Chemistry at the University of Cambridge, and I am a fellow at Corpus Christi College, where I'm a Director of Studies for the chemistry-focused undergraduates, and I'm involved in the small group teaching, the supervisions for the organic chemistry courses in all years. Of course, I've not always been in Cambridge, so I'm actually from the Southampton and Portsmouth area in the south of England. And then when I started my chemistry career in 2010 on my undergraduate degree at the University of Birmingham, I've since been all over the country and also to many places around the world to work and to study. So chemistry really can take you globally if you wanted to. So now back to some actual chemistry. So I am an organic chemist, but what actually is organic chemistry? So I hope you understand from your A-levels that organic chemistry is one of the three major branches of the very nebulous term chemistry. And organic chemistry specifically focuses on studying the structure, properties, and reactions of molecules that contain carbon, otherwise known as organic molecules. And such organic molecules can be very simple ones, such as hydrocarbons, methane, ethane, propane, and so on. But they can also be much more complex and feature non-carbon-based groups, such as in these molecules, aspirin and paracetamol. And you're perhaps familiar with these from your studies at school, or perhaps you've taken them as a medicine yourself. But organic molecules can get quite a lot more complex, such as with morphine here. This has a relatively complex structure with many different rings and different non-carbon atoms. And as we shall soon see, there's almost no limit to the complexity of molecules that can be designed and built by organic chemists. So now we know what organic chemistry is, why should we care, why is it important? So firstly, the principles of organic chemistry are obeyed by all biological systems, with biological systems, in my opinion, really just being very complex sequences and cascades of reactions of organic molecules. And so our understanding of organic chemistry is essential to our understanding of biology. And in addition to this, organic chemists are involved in the development of tools and methods to study and increasingly influence biology. And so next, have you ever taken a look at the ingredients on the back of a shampoo bottle or a cleaning product or something else? Well, a lot of those names that are very difficult to pronounce are in fact organic molecules, and they're called specialty chemicals. And these are essential to a huge range of industries, including like agrochemicals, so things such as pesticides, herbicides, and fertilizers, but also food additives, flavorings and colorings and preservatives, fragrances for perfumes and paints, just to name a few. And one very large subset of such specialty chemicals are polymers and plastics, and the vast majority of these are simply very long chain-like organic molecules that are comprised of repeat subunits of smaller organic molecules. And of course, plastics have permeated pretty much every part of the modern industry globally, and so they're very important molecules. And finally, the vast majority of drugs and pharmaceuticals are organic molecules, and these are all prepared by organic chemists. And of course, these molecules are of quite fundamental importance to our lives. And without this industry and our understanding of organic chemistry to support it, people would still be needlessly suffering and dying of diseases which we now consider to be very easily treated. And so for the rest of this talk, I'm going to continue within this theme and discuss in more detail two brief case studies on the application of organic chemistry to challenges in the drug and pharmaceutical industry. So we're going to take a look first at this organic molecule called Taurusin. And this structure is probably quite a bit more complex than any molecule you've seen at school. But it's a sort of structure that organic chemists, especially those developing drugs, become quite familiar with, as it contains many features that are very common in drug molecules, including many atoms other than carbon and hydrogen, in particular nitrogen and oxygen, but also some fluorine atoms as well. It also has many functional groups, such as carbonyl groups, aromatic groups, and it's got chiral centers. And I think all of those you should be learning about in your A-levels. And so this molecule itself was only approved last year, and it's considered to be a milestone drug for the treatment of advanced lung cancers. And why is it a milestone? It's because it can target a enzyme called KRAS, which up until this point with this molecule was considered to be undruggable for various reasons. And what's even more special is this molecule can target a specific mutation to this KRAS enzyme that is prevalent within specific tumor types. And so because KRAS acts as an on-off switch for cell proliferation, if we can target the mutated KRAS within a tumor, we can essentially force it to be in the off position and therefore prevent further tumor growth without affecting the healthy cells. So another interesting point to note, which I'm going to be focusing on today, is the fact that this drug is used as a single A-trop isomer. And this is essentially an unusual form of stereochemistry that I'll be discussing shortly. And so several years ago, this molecule actually looked like this. And you can see it has many similarities to the final drug, Taurusin, but there are many notable differences too. And so these differences are highlighted here in purple in the molecule on the right-hand side, which is just Taurusin with some highlights. And so the change from the original molecule to the final one was actually the result of several years worth of work by dozens of organic chemists who were charged with doing a variety of different things. So predominantly improving the potency, selectivity, and solubility of this molecule, as well as improving its pharmacokinetics. So in brief, that means what the body does to the drug and vice versa. As part of this process, chemists were also responsible for making sufficient material to be put into the various tests. So that would be biological assays where you're doing tests on isolated proteins or in cells, animal studies where you're normally testing for safety as well as a few other things, and then the final clinical trials when the drug goes into humans for the first time. And at that point, you need quite a lot of this drug. And then, of course, if the drug is successful and gets approval, then organic chemists will be making this drug on a very large scale so that it can be distributed globally or wherever it has been approved. And so now, let's take a look at what I mean by A-trop isomerism and why it is important and relevant to this drug and its development. So I hope from your A-level studies that you'll be familiar with the concept of stereocenters or chiral centers. And I've depicted this in the box here. So for those that haven't been taught this yet, for a tetrahedral carbon atom, if you have 4 different groups attached to it, it can exist as 2 different enantiomers, which are defined as non-superimposable mirror images of one another. And so for the molecule shown on the left-hand side, which has 4 differently colored balls attached to a central tetrahedral carbon atom, if you reflect that molecule through the central mirror plane to get the molecule on the right-hand side, that reflection then cannot be moved or rotated in any way to map it exactly back onto the original molecule. Therefore, these molecules are different and are called enantiomers of one another. This is regular stereoisomerism. And so what about A-trop isomerism then? Well, there are actually other forms of chirality that are similar to regular stereoisomerism, and A-trop isomerism is just one of those. And so for this case, if we have the same 4 colored balls that are arranged in exactly the same way, but rather than linking them through a tetrahedral carbon atom, if we instead link them by 2 bisecting planes or lines that are linked together somehow, the result of that is in fact chiral in the exact same way as before, in that if you take a mirror image, that mirror image can then not be superimposed back onto the original. So these two will be called atrop isomers of one another. So then how does this relate to the drug Taurusin? So let's take a look at the structure in more detail, and in particular take a look at the section I've highlighted in purple. As you'll see that on the aromatic ring on the bottom right-hand side, you'll see I've drawn the aromatic ring a little bit strangely. So those bold lines that indicate that that edge of the ring is actually protruding from the plane of the screen towards you. And so that ring is actually perpendicular to the other purple ring on the left-hand side. And let's take a look at that by removing all of the redundant parts. We can now see how this is beginning to look like two bisecting planes. And it's linked by the bond I've highlighted in red. Now, these can only truly be atrop isomers if there is restricted rotation about that bond I've highlighted in red, which in this case, it is highly restricted. So if we now put that model into the cartoon from above, we can now clearly see how this section of Taurusin has atrop isomeric qualities. And so hopefully now we understand what that means, but how does this relate to the molecule and how does it work and why is it important? Well, the answer is that this particular atrop isomer, as drawn on the slide, is critical for its potency. And so if you remember back a few slides ago, I said that the story of this drug started off with a different molecule, this one here. And so one of the first things that organic chemists did was identify that putting an aromatic ring, which I've shown in purple, in that particular position, increased its potency 20-fold relative to the original molecule. And this is also the point where atrop isomerism was first introduced into the molecule. As you can see, I've drawn it in the same way. However, what was found is that at room temperature, that red bond can actually undergo rotation, albeit quite slowly, to give the other atrop isomer, which is unfortunately only 2 times as potent as the original molecule, and of course, only 1/10 as potent as the previous atrop isomer. And so in order to maintain the high potency, they needed to somehow prevent that bond from rotating. And so to lock that bond, what organic chemists did was that they placed a methyl group, as shown here in purple, so CH3, to prevent that bond from rotating. So that group is much larger than the original proton, so the original CH, and that locks it and prevents it from rotating. And so that maintains its high potency, but also in this case, in fact, doubles its potency to being 40 times as potent as the original molecule. And now this really isn't very far from Taurusin itself, so we just need to do a final few additions and a few modifications to the structure, and we get to the final molecule shown on the right-hand side, which is Taurusin, and we can see that this is 80 times as potent as the molecule that was originally discovered. And so that concludes the short story of Taurusin that I'd like to discuss today. And so for the final few minutes of my talk, I'd like to go to the story of an even more complex drug molecule called Ribulin. So Ribulin has this immensely complex structure with many oxygen atoms, many rings, and many chiral centers, all interwoven into one another. And so this molecule was approved in 2010 for the treatment of some of the most aggressive and extensively pre-treated metastatic breast cancers and liposarcomas. But whilst that's obviously a really great outcome, the reason why this molecule is so revered by organic chemists is that it is the most structurally complex, fully synthetic drug on the market. And this is in stark contrast to many other very complex drugs, which are instead extracted from natural sources. So such as morphine that we saw on an earlier slide, which is in fact extracted from poppies. And so to put the synthetic challenge of this molecule into context, this has 19 stereocenters or 19 chiral centers. And so if we consider that there are actually 2 possible forms of each of these chiral centers, there are in fact over 500,000 possible combinations, and organic chemists must be able to navigate all of those and make just one of them, which is the final compound. What's also interesting is that this molecule, the structure, was not simply pulled from thin air. Instead, it was inspired by the structure of this molecule called halachondrin B, which was isolated from a marine sponge and also has very potent anti-cancer activity. So this is what we would call a natural product because it is isolated from a natural thing, a marine sponge in this case. And so chemists sought to synthesize this in the lab, and that is a process called total synthesis because it's synthesized in its entirety from simple pieces. And during that process, some of their intermediates were tested for their biological activity, and they found that the purple section of halachondrin B on the left-hand side was almost essentially redundant and not really required for its potent anti-cancer activity. And instead, it could be replaced by a relatively simpler piece, shown in purple on the right-hand molecule. And then, following a similar process of optimization to what I described for sotorasib, the molecule is then optimized from the one shown in the bottom right to give eribulin in the top left. And so while eribulin is obviously incredibly complex, halachondrin B, the original molecule, is far more complex, and it has 32 stereocenters. So going from 32 to 19 is not quite a reduction by half in terms of the raw numbers of stereocenters. But if we consider that with 32 chiral centers, the number of possibilities of orientations of all those stereocenters is almost 4.3 billion, we're actually doing an 8,000-fold simplification by going from halachondrin B to eribulin. And so now the big question is how is eribulin made in the laboratory, and how do organic chemists like myself take such a complex molecule and come up with a way to make it? And so one of the key skills of an organic chemist is their ability to do a retrosynthesis, which simply means backwards synthesis. So how can one take a complex structure like this and go backwards in the reverse synthetic direction to get much simpler starting materials from which we can construct this molecule? And therein lies the beauty and challenge associated with making such molecules, and why total synthesis is considered to be an art form. Because we must be able to take a limited number of organic reactions and a limited number of chemicals available and make something as complex as this. And so for eribulin, for example, we can actually break it down to get much smaller molecules. So for example, I have extracted this fragment here, which is shown in multiple colors. This actually corresponds to the left-hand side of eribulin. And you'll see I've colored it in various parts. Well, that's conveniently to show that they correspond to several different molecular building blocks, which we can buy from suppliers. So we can effectively use these like Lego and construct them and stack them all together to make something much more complex. So if we take a look at that in a little bit more detail, when you have something very complex like eribulin, the best way to break this down is to break it right down the middle. And so in this case, if we break the bonds shown in red, so we're going to break a carbon-carbon bond at the top, we're going to break a few carbon-oxygen bonds at the bottom right, and also exchange the nitrogen group for an oxygen group. We get back to these two smaller molecules, which are looking now a bit more simple, though still quite complex, but they're about half as complex as the original eribulin molecule. And so then how do we take this further and to make even more simple molecules? Let's now take a look at the left-hand molecule. And so again, if we break this approximately by half, we break some bonds in the middle. We're going to break a carbon-carbon bond and a carbon-oxygen bond shown in red. We then get back to these two smaller, simpler molecules again. And this is now beginning to look a little bit more accessible. And hypothetically, we can keep going and going until we get back to those very simple building blocks I'd shown on the previous slide. We don't have time for that today. And so what we will do, though, is take a look at the small molecule on the right-hand side, break it once more at the bond shown in red to give us these two smaller building blocks. And I'll talk through how we make the bottom one in the laboratory. And so if we start off with these two colored building blocks, which I'd shown on the previous slide, we need to do some reactions with them in order to eventually link them together to make this desired molecule. And so for the top pink building block, what we need to do is react the carbonyl group to get an alkene in an olefination reaction, and react one of the alcohol groups to introduce a bromine atom in a bromination reaction to get the molecule on the right-hand side following those two different reactions. And so for the building block on the bottom left, the blue one, what we need to do is react one of the alcohol groups to form an aldehyde, and that will be called an oxidation reaction. And now what we can do is link those two molecules together through the aldehyde and the bromide. And these can react together in a process called alkylation to form the molecule shown in the center of the slide here. And now we can clearly see how we've now linked together those two original building blocks. The next thing we need to do then is link together those two highlighted alcohol groups and do a cyclization reaction, so a substitution we would call it, to form a 5-membered ring which contains the oxygen atom. And so the last thing we need to do is just take that final free alcohol group and convert that into an aldehyde. We've seen this before in the blue molecule previously, as that's just another oxidation reaction. And all the reactions I've described here in very brief terms are reactions you will learn about in some of the early undergraduate organic chemistry courses. And so that brings me to my final slide, which is a summary of everything I have discussed today. So in the first part of the talk, I discussed some aspects of this groundbreaking anti-cancer medication called Taurusin, which I hope functioned as a bit of an introduction to medicinal chemistry, which is how organic chemists design drug molecules. And I also focused on a particular aspect of this molecule, which was the atrop isomerism. Following that, I then discussed this very complex molecule, Ribulin, which I hope functioned as an introduction to total synthesis and how organic chemists can construct these complex natural products. And I also had a particular look at the concept of retrosynthesis, which is how organic chemists can deconstruct these complex molecules on paper to design a way to make them in the lab. And so with that, I thank you all for your time and for listening to me today. I'll now pass over to my colleague, Adam Clancy, for the second part of today's afternoon talk. If you do have any questions, please do make a note of them in the Q&A, and then I can address them either while Adam is talking or after he has concluded.
Cool. So
over to you, Adam.
Can you hear me?
Yes.
Yeah, brilliant. Yeah, now it's to challenge, make sure the screen share works. Oh, it's on the wrong bloody one. Do you want to know how to reverse this?
Click on the dots, I think. The left, down, left and down. There's like 3 dots. Maybe it's that one. It's one should be swap the presenter view. Click on that one. Click on the screen one to the left then.
It's hide presenter view. If you click on hide presenter view, that should work.
I don't know what that is. That one. No.
Go back to here. And then go. Wait,
that just turns it black.
Oh no. Okay, maybe try this one.
Oh well, I'm just going to stop sharing and just share the other screen. It's for easier. Share screen 2. And then that work?
Yes, that's working.
Yeah. And can you see the pointer? Cool. Okay, so my name's Adam. I'm not actually from Cambridge originally, so I've been all over the shop. So I actually did my master's and my bachelor's in Cardiff in chemistry, then in my PhD in materials in Imperial College, and now I work mainly at UCL, but also have got a visiting fellowship in Cambridge. So I've been all over the shop. So my research itself focuses on materials and nanomaterials. So I tend to dissolve them, change the surface properties, and then assemble them. But if you have any questions about my research, then you can Google me, or my email's at the bottom. Someone's asking for this in the chat earlier, and got Jenny's. But feel free to ask me any questions about nanomaterials in general or my research. But today I'm just going to talk about nanomaterials at the very basics, because this is something you may well have seen on BBC or sort of read in science fiction, but really understanding what nanomaterials are tends to be something that comes later in a chemistry research degree. So try and give you sort of a bit of a head start on that. So first of all, let's talk about what the nanoscale is. So this is just a logarithmic scale down from 1 meter down to 1 picometer. So this is as big as everything you can hold down to the smallest things we can physically see. So trying to find an example of something which is 1 meter big as a guitar. If you want to go down to sub-centimeter, so 5 millimeters is the width of the eraser at the end of a pencil. And smaller than this, down to around 70 microns, is the width of a human hair. And this is about the limits of what a human eye can see. But obviously, there's a lot smaller things on this. So if you look smaller again, you can get down to sort of red blood cells at a few microns wide, a micron being 1/1,000,000th of a meter. Smaller than that is something we're all very acquainted with, unfortunately. So viruses are on, tens to 100 nanometers wide, and around this range, so viruses touch and go. To see them, you need to use a light microscope. And then even smaller, we get to the width and the length of DNA segments, just above a nanometer. For this, you need even more advanced microscopes, and TMs are something becomes very important for materials chemists like myself. And then eventually you get down to atoms, which is the smallest you can go. So this is basically the entire length scale of matter on the planet. For that, you need even more specialist microscopes. You see they start getting bigger and bigger. You just look at the size of the chairs compared to the microscopes themselves. But the region I care about is this area, which are boxed in green. So about half a nanometer up to about 20 nanometers. Because some weird and wonderful things start happening there. This is quite weird because most chemists tend to work a little bit to the left of this green area where molecules are, which sit in between your large DNA molecules up and your single atoms. So why do I care about this green box? And it's because it's where the behaviors of things start changing. So if you take a lump of gold, I personally have never seen a large lump of gold, but I'm sure that this is what it looks like. And you cut it in half in a sort of Plato-esque sort of way, you end up with 2 lumps of gold, and they behave exactly the same. When you cut it in half, it starts as 1 lump of gold, which has the properties of gold, and then it becomes 2 lumps of gold, which, while smaller, have the properties of gold. And you cut it again, and you end up with something smaller again. Cut it, and you keep going, and eventually you'll cut it down so that you've got single atoms, which do not behave like the rest of gold. So there's got to be something in the middle where it transitions from behaving like the bulk material to behaving like single atoms. And this is where nanomaterials come in. And the sort of weird behavior you start seeing when you start getting into this regime can be split into many different properties. The 2 to focus on are optical properties, so how these behave with light. So for example, this one, the first known examples of nanomaterials, although it was made by accident, which is a cup from the Roman Empire where it has gold nanoparticles distributed within glass. And this means that gold nanoparticles give this sort of reddy color. They don't have this sort of gold sheen of gold blocks. The properties are completely changed. When you shine a light on it, it'll bounce off and it looks blue. Shine a light through it, and it absorbs the blue light to give you this sort of red background. So these weird optical effects. And these sorts of optics will also change how you can use these materials in solar cells or catalysts, and a large number of applications moving forwards. And the other thing which nanomaterials are known for is that they're really high strength. This is actually probably I'm going to focus on a little bit more within this talk, with the classic question being, with the strongest materials in the world, carbon nanotubes, which we'll focus on, can you make a space elevator? And the answer is, I don't know. No one's ever done so successfully, but it's about as good as materials we can hope to give a good shot with. You need something incredibly strong, and you can probably get away with it until the very difficult conditions of space destroys it and it comes crashing down. So as examples of nanocarbons, we can split them into different families based on what we call dimensionality. And this is, when we say it's got a nanometer range, this can be in all dimensions. So if you take your XYZ coordinates and you look and you say everything will be within a nanometer in all of these directions, it is what we call 0-dimensional, or typically called molecular. These are just molecules. These are fake nanomaterials. They don't really count. Instead, what you can do is start with something which is limited only in 1. So if you take graphite, a 3D material, you've got pencils which have graphite in them. It's a large thing you can hold on to, definitely 3D. This is infinitely long in all of the directions. By infinitely long, I really mean more than 100 nanometers, and after which it's just the behaviors don't change when you get bigger. But if you limit that to a single sheet, you get something which you may well have heard of called graphene. And one of the questions at the end, which I've been referred to, it does have is about graphene. So it's very well known. And this is a 2D crystal where it's limited in one axis, but not limited in two. So it can be infinitely wide, infinitely sort of flat moving forwards, but only one atom thick. And this you can then roll up into your zero-dimensional material, or you can take a sheet and roll it across in one direction to leave one direction, which is infinitely long, and this is your carbon nanotube. So this is infinitely long in one direction, nanometer limited in two of them. So this is a 1D nanomaterial. So why do we care about these nanomaterials? Well, it's the applications. It's the fact that these can be used for thousands of different properties, lots of different applications based on their amazing properties. So they're used in solar cells. They can make aerogels, which are effectively as light as air, but can start supporting quite strong weights. We can use them in transistors for computers. I've personally used them to grow proteins off them. Other people do use them for things like invisibility, which is meant to be a video if this is going. So this is a real effect, a real video, which is playing with the thermal electronic properties to make it disappear and sort of bend light around it. So when you have these nanotubes, there's lots of different sorts you can have. So when you buy them from your supplier or you make them, you get what is indistinguishable from just black soot. What's quite neat is that because these are made by wrapping your graphene sheet, so you can imagine having this little square of graphene, you can sort of wrap it along this red edge and then follow the red arrow and you get a nanotube which is like this, or you can wrap it across the blue, these blue lines, sort of bond these carbons together, and you get something which looks completely different, both from the side and looking down it. So we refer to these particular two extremes as zigzag based on this sort of pointy edges on the top one, or armchair based on this armchair configuration going down the edge. Or you can just pick a random carbon and link them across, and you end up with a weird spiral effect. These are called chiral. Now, if you can think of it as linking any two particular carbons, you can sort of draw the line between them. So this is going along here for the blue, or down here for the red. If you sort of draw this across a graphene thing, you can get very specific things called chiralities. And this is every individual nanotube which can ever exist based on carbon. And interestingly, while we do have this black soot to start with, that's just because we've got a mix of everything. So each of these individual ones, so this one would be a zigzag, so we'll end in a 0, which we don't actually have an example here, annoyingly. Or we've got this one here, which be an armchair, and they all have different properties, different colors. So they absorb light in different ways. They behave in different manners. So you can use them to make different transistors. You can make them to make better solar cells. And it's only when you combine them up that they become this sort of mix of black. Now, this is something which is not sorted. If someone could do this on an industrial scale, this is when we'd see real big changes in society and materials and devices. The problem is this can only be done at the moment with tiny, tiny amounts through a very difficult and arduous process. So hopefully by the time you've gone and done your degree, in your 4th year, you do a research project, you can easily be envisioned being given this problem and told solve it and separate all these nanomaterials, all these into the different colors, and try and do it in a way where I can give you a kilogram of nanotubes and you'll separate them out. These are the sorts of problems we're tackling in material science. Let's look at one of the main things which people remember about graphene and nanotubes is talking about them as some of the strongest materials known to man. So I might delete my K on stilt, but if you compare to some of the other great materials in the world, so steel or Kevlar or spider silk, which is sort of one of the toughest materials known to man, and then you plot the properties against each other, you see that the strongest thing is carbon nanotubes. And this is how much stress, how much energy do you have to put into a material to snap it. And the strongest thing is nanotubes. There's the modulus, which is if you pull it a very small amount, what's the gradient of this sort of, as you pull it, how much energy do you have to put in? And the stiffest thing is carbon nanotubes. for the record, when people are making composites and the fibers for materials for Formula One cars, people don't really care about the strength. It's actually the stiffness that they care about, but it doesn't sound as impressive. And finally is the toughness, which is if you basically keep pulling it until it snaps, how much energy did you have to put in? So this is representing these graphs as the area underneath. And again, the answer is it's nanotubes. Nanotubes are just the strongest, stiffest, toughest materials known to man. And they beat all the competition on every metric. And this is seen in most nanomaterials. The only thing which is comparable with a nanomaterial tends to be another nanomaterial. So you might ask why being small makes something stronger. And this is actually quite simple if you sort of think of the physics behind it. So the formal version of it is the strength of material times by the cross-section, the root of the cross-sectional area is approximately equal to a constant which varies by material, which effectively means if you make something smaller, the strength goes up, which sounds weird, but is definitely true in macroscopic materials. And this works even down to the atomic level. So to think of it with the really simple model, everyone's heard the phrase, a chain is only as strong as its weakest link. So what we really care about is the weak links in our system. So if we take this chain, this happens to be 30 units long, and we say there's a 5% chance of a weak link, what's the odds that we actually have one weak link or two weak links or three weak links, or however many? And you get this equation, which I've written wrong, so there should be a factorial after this, n minus x. But you can literally get the probability. You can see that it's most likely there's going to be one weak link, and there's a decent chance of getting two, etc., etc. If, however, you make the chain shorter, you make it smaller, then suddenly you're just as likely to not have a weak link as having a weak link. And if you make it even shorter still, going to 10 links, then it has basically, it's more likely than not, it's 59.7% likely to have no weak links in your material. So when you start talking about materials in general, we have what we call defects, and defects are what really determine how weak something is, or how limited the strength is versus a perfect material. And as you go smaller, you get fewer defects and your strength goes up. So your nanomaterials are always going to be stronger than your non-nanomaterials. So if we've got what's allegedly the world's strongest material, I want you to just take a moment and think. So I've given you all the information required for you to do the maths, and we'll work through it together in a second. How strong is the strongest material? If you take one nanotube and you hang a weight off it, how heavy does that have to be to break the strongest material known to man? So I've given you 6 examples. Could a nanotube support a human cell? Could it support an eyelash? If so, could it support a paperclip? Could it support a mouse, which is apparently 21 grams according to Google? Could it support a pineapple? Also, if no one knew that pineapples grow on the tops of these things, I didn't either. Or could it support an elephant? I'll give you a clue, it's not that one. So if you're with someone, see if you can get them to sort of guess as well. And if not, just have a think. This is the strongest material in the world we're talking about. So, got your guess? So, very simple. This is GCSE physics when you think about it, really. That we have force equals mass times acceleration. This is the force which the weight is going to be applying to us. And then we've got the strength of the nanomaterial, which is the strength of force divided by area. So you can combine those together and sort of move it around to give you a mass. And we know that our strength is 60 gigapascal, which was on a graph earlier. We know our radius is 1 nanometer, so the area is going to be 7.85 times 10 to the minus 19 meters. And our acceleration is gravity. So we just need to work out what the mass is. So you just plug it all in, and you find out that it's not even an eyelash, it's 1/10 of an eyelash. So the strongest material in the world is 1/10 of an eyelash. Hanging off it is enough to break the strongest material in the world. And the issue is that we're only looking at one of them. And while it's a benefit to have tiny materials, these nanomaterials which have few defects, incredible strength and optical properties, the issue is they're very small, so you need lots of them. So how do we assemble them into something which can actually support, support an elephant? So to do this, there's a couple of things which can be done. So there's a beautiful, elegant work of when you grow nanotubes, they come to these sort of what we call forests. They grow vertically off the surface. If you just sort of pull at it, and as you do so, pull and twist, you can turn it into a fiber. And these things do have the strongest strength, the highest strengths of anything known to man. Something you can actually hold in your hand and try and pull apart, and you will fail to break it. Or alternatively, there's another route, which is, this was invented in Cambridge. This was by a professor called Alan Windle several years ago. So what he does is, as you make the carbon nanotubes, it forms like this smoke inside your furnace, and you can take a really long pole, stick it into the furnace, and you can grab the smoke. And the same way that these ones tangle with each other as you pull it, but you have to start with this nice forest. Here, you just grab the smoke which is growing and then pull it out like candy floss. So these are the routes of assembly. So there's another major area to investigate. So turn these things from individual strong materials into something actually practical. Lost time to stop sharing. So, Jenny's.
Yeah, thank you so much, Adam. I guess, James, would you like to referee some questions for us?
Yeah, can we all, I mean, I want to know, can we all go to the lab and help you build nanomaterials? I mean, yeah, let's do it. Let's organize a field trip. And also to the person who asked about material science and what it is, and I desperately vamped and didn't know the answer. I think you've there just got a pretty good indication of what material sciences at Cambridge is like. Do we have any, okay, so the first question is it possible to predict the weird behavior of materials we know normally at the nanoscale?
Yes, absolutely. It can be quite challenging, but it tends to be a lot of people with computers doing it, and then they go away, tell you it should do this, and then so I'm an experimentalist, it's my job to actually prove that it works, but they absolutely do this. There's a whole branch of chemistry called computational chemistry, which is a huge part of which is trying to understand materials. So not only is it taking materials we do know exist and predicting their properties, and explaining why these things behave like they do and how we can potentially take advantage of it to, revolutionize solar cells. You can even take it a step further and say, okay, this material doesn't exist, but we reckon we can show that it should exist and it should have these properties, and they should even be even better at revolutionizing the world. And on a personal level, I hate the person who comes to me and says, can you make this brand new material? But you absolutely can. The limits of computational chemistry is virtually, endless. It's a very powerful tool, and it's a reason why it is. I mean, Alex described chemistry as having 3 areas. So he's organic. I would describe myself as inorganic. Physical tends to be measuring. And then as a sort of off branch of physical and organic and inorganic, there's kind of this 4th computational chemistry, which particularly moving forwards is going to be one of the biggest fields. And it's very, very much a growing one.
Could we add on that, James? Just because we had a previous question that asked about how computational chemistry would also change medicine. I wonder whether we could ask Alex to help us out there with that question.
Yeah, sure. Well, obviously I am not a computational chemist myself, but my fiance is. And so she works in a pharmaceutical company as a computational chemist developing drugs. And so, yeah, it's been around for quite some time, and it's increasing in its power to do things. So when it comes to medicine, computational chemistry can do various different things. And so primarily I would say there's one triage, in the sense that if you gave me a potential drug molecule and asked me to optimize it to make it a better drug molecule through various parameters, I could come up with 100 ideas, but I'd have to make them all in the lab in order to know which ones are good and which ones are bad. And that's incredibly time consuming. But a computational chemist can model those proposals inside the enzyme I'm trying to inhibit, and then go, actually, those 10 are really the only ones that look promising. And that can save me in the lab a huge amount of time. But also what they can do is they can try and propose entirely new things. So for example, perhaps you've got a crystal structure, a structure of an enzyme you want to inhibit, but you don't know what molecule can go inside and inhibit it. Well, computational chemists can perhaps model and insert structures in there and see what can happen, without having, again, to have somebody in the lab spending an incredible amount of time, literally trial and error, just to see what works. And so they're probably not the only things either. There's also data analysis. So you could, for example, take a huge data set of activities of various drugs against a particular target, and then with that data model what you think would be a good modification to your drug. Yeah, there are many aspects, and it's going to have huge impacts, I'm sure.
There's a question here from Emma, which I think possibly is for you, Alex, again, so you unmute yourself quickly. So when you have synthesized a molecule, how do you actually check that it's the one that you wanted to synthesize in the first place?
So I started typing an answer, but I realized it could also be to Adam as well. So because Adam also has a problem making things and trying to figure out what they are. And so in terms of organic chemistry, the answer is spectroscopy. And so NMR spectroscopy in particular is used. And that's something that you do learn about in your A-levels. So if you haven't done that yet, it's probably going to be this year that you learn about that. And that's an incredibly powerful technique for working out what you've actually made. I don't know about Adam, so perhaps Adam can jump in and say, if he's making his nanomaterials, how does he characterize those?
So spectroscopy is a huge one. I do something called neutron scattering as well, which involves shining a beam of protons onto a lump of tungsten. It then falls apart, releases a load of neutrons, and as far as I'm aware, I just basically put my material in the outside of it. But it lets you sort of see where hydrogens are in things, for some of my hydrogen-containing stuff, and getting liquids in. So spectroscopy is the main one. We do have more elaborate, exciting ones, but those are the primary routes. So how things interact with light and how things react with neutrons.
I can also see that Adam, you were typing an answer. I think that'd be. I mean, I also want to know the answer. So if you could talk about that live, that'd be good.
Yeah. So I was trying to. I didn't know you could see that I was typing. This is the question: if you assemble the strongest materials, which are tiny, into a big object, does this reduce the strength? Well, you kind of caught us there. Yes, absolutely. The thing is that you need to do so in a way which limits the number of additional defects you put in. So when you assemble it, you sort of have these nanomaterials touching each other. So if you take graphite, the reason that's weak is it's got made of graphene, which is the other contender for strongest material in the world. But graphite, your pencil, is so weak that if you lazily pull it across a piece of paper, it will start degrading and falling apart and leaving these graphene sheets on your paper in what we call using a pencil. But what you need to be able to do is, that's because you've got these really weak interactions between these materials, which even though they're the strongest material in the world, how do you make, when one moves, how do you make sure that the other one's pulled with it? So with the carbon nanomaterial fibers, we twist them so they get tangled together like a rope. And therefore we can move the sort of stress put onto one onto the other. And you can do this in a way which is a lot more effective than other ways of assembling it. And we start with something which is fundamentally stronger to start with. But yes, when we say we make something 60 gigapascal strong, which is incredible, to my knowledge, the best we've managed to sort of maintain of that is 8 gigapascal. So we've lost almost 90% of the strength, but this is still 10 times stronger than pretty much every super strong material known to man. But yes, you are right. Good catch. We do make it bigger. It does become weaker.
And this, just to go back to computational chemistry, I'm not sure who'd be best placed to answer this one, but so a couple of people like to know, how do you actually get into computational chemistry?
Do a chemistry degree. It's a huge part of it. You'll be doing your in your first year. They didn't tell me I was learning quantum, which I felt a bit cheated by. But the second you start hearing the word eigenvalue, this means that you're being taught computational chemistry. And you just need to keep sort of following that. If it's something which makes a lot of sense to you, or makes any sense to you, you'll be ahead of the curve. And it's sort of do a chemistry degree and then specialize in like your master's year for your research project.
I would also add to that and say, if you are seriously interested, consider just taking up some regular programming classes or learning on your own. I'm not really the person to ask, but I think things like Python are going to be useful. And so it's not like using an app on your phone where you can just click things and model this or whatever. It really is hardcore programming sometimes. And so you do need programming knowledge in order to do well. So you obviously get the chemistry from the chemistry degree, but the programming is still ultimately programming you have to learn somehow.
Yeah. I'm conscious that we've gone past the time, but are you happy to answer a few more questions, or have people got to go off? Oh, okay, that's great. You regret that you agreed to that. Now we'll see another like 50 questions come in. This is from Alexander. I think it's for you, Adam. Aside from the obvious application of construction, what would you say is going to be the most impactful application that carbon nanotubes might have?
Transistors.
Good question.
So the mechanical side of things, we already use nanotubes. So they're used quite a lot in tennis rackets, for example, because they're quite easy to. When you make carbon fiber ones, they're very brittle, they break very easily. And carbon nanotubes, it's not so much that they're strong, carbon fiber is already very strong, but they're really tough. So it's just to sort of mitigate them sort of shattering when they're broken. So the mechanical side of things are already used. They're also almost certainly used in phone batteries at the moment, although we can't technically say. It's just very weird that all of the phone manufacturers happen to have built carbon nanotube battery factories, carbon nanotube synthesis factories, and then when people go, are they being put in your batteries? They go, no, I don't know what you're talking about. So that's on the industrial side has driven that. But for me, the thing which will be amazing moving forward is transistors. So 2/3 of carbon nanotubes are semiconductors. They've got properties which make them so much better than silicon in every way, apart from the fact that when we make a silicon transistor, your microchip, you take 100 million circuits and you put them in an array and they all work 100% of the time. And we don't have that reliability for nanotubes. Once we get the reliability, and we can make sure that when we put 1 million nanotubes down, they all work perfectly, there's going to be a jump, a huge jump. They're cheap, low energy to make, and that will be the next big jump in electronics generally, not just carbon nanotube science, in my opinion.
How far away are we from that, do you reckon, Adam?
We've been 5 years away since they were discovered 20 years ago.
Okay, so let's not hold our breath. You should be in the lab then. Get off this call, get working.
I'm off for the week. This is right. I have as many questions as necessary.
I think this might be a question for all three of you, actually. I'd be interested to get your opinion on it. How do you think sort of the development of AI will affect chemistry in the future, or affect it now, but also in the future?
So one thing, okay, again, my perspective is organic chemistry. So one thing that AI is perhaps going to be useful for in the future is the process that I described in the second part of my talk, which was the retrosynthesis, taking something very complex and reducing it down to simple things that you can then build from. And so people are trying to come up with programs that can do that, but it's incredibly challenging. And so people are trying to incorporate machine learning and AI into that process by taking all of the published work from hundreds of thousands of people around the world, somehow crunching that into some model, and then using that to propose syntheses of very complex molecules and drug molecules. So the end product could be somebody in a pharmaceutical company goes, I want to make this, and then the AI just says, here are the procedures, do this. But soon that could perhaps even be combined with an automated technology that could run everything for you. And so from beginning to end, it's just robots designing and doing.
We all sign on to the dole.
Exactly, yeah.
The question for Adam, and actually I'm glad that someone asked it because I wanted to know this as well. You talked about growing carbon nanotubes, and someone would actually like to know, how do you do that?
So you start by, there's different routes, but the most common way by an absolute mile now is you take a small metal nanoparticle which can dissolve carbon really, really quickly. And then you take a carbon gas, normally something called acetylene, which is carbon with the triple bonds, 2 carbons with triple bonds and hydrogen either side. So very very carbon rich, 50% of your atoms are. And the carbon dissolves in your metal, and it keeps doing this and breaking down until there's too much carbon dissolved and it starts precipitating out the other side. So you have a nanoparticle about this big, and then you make a nanotube also about this big, because your sphere just sort of gets coated on one side by a bit of carbon and then just starts growing out as the sphere stays in the same side, and carbon comes in one end and it gets kicked out the other. You can do that by putting the nanoparticles on a surface. So when we saw this forest earlier, they're forced to grow up, because if they grow sideways, they bash into another nanotube and then have to go up anyway. So that's how you grow these sort of forests of nanotubes. But you mainly nowadays just sort of like swirl them in the air and react to them that way.
So we use the one with the furnace and catching the smoke rather than the growing.
Yeah, so that furnace is sort of is the sort of nanoparticles sort of blustering around and then sort of growing these really long nanotubes. The nanoparticles this big, which it grows out of, and then you've got a nanotube which is this long. There is actually an issue trying to then get rid of the metal afterwards, which I've personally done some research on. But then there's sort of like the sort of wavy bits of string floating in the air, and that's where you can sit with the furnace, just go yoink, grab it, and then start twisting it and sort of catching these nanotubes, which are growing as this sort of tangled smoke.
Wow.